we know that in addition to TCRs, there are many surface antigens that further define the identification and functionality of T cells, giving them distinctive characteristics and directing their interactions within the immunological cell communication. And these surface molecules that are found on other immune cells, in addition to T cells, were identified and characterized by the monoclonal antibodies. And these monoclonal antibodies are the laboratory-produced antibodies produced by cloning a particular cell lineage that have the ability to bind only specific epitope. So particular type of monoclonal antibodies were designed to recognize specific cell surface molecules. And thus, they were used to study and distinguish different cell populations within the immune system, allowing for a standardized way to classify and describe them. This method is one of the most important advances in immunological diagnostics. And thus the molecules that were identified by this technique are given the specific designation internationally. And that is CD, or cluster of differentiation. And these CD proteins, short for cluster of differentiation proteins, are a classification system used to identify and categorize cell surface molecules found on various immune cells, including T cells, B cells, natural killer or NK cells, and others. This CD nomenclature assigns a specific number to each protein, like CD designation at the beginning followed by a number, indicating its presence or absence on different cell types or stages of cellular activation. Thus we can say, the surface molecules on immune cells are actually the cluster of differentiation proteins, or CD proteins. And if we talk particularly about T cells, then those differentiation proteins that are found on T cell surface are called T cell antigens. So, now let's look at these CD molecules in more detail. But there are over 370 recognized CD proteins, so we will not discuss each of them. But we will look at those that are particularly related to human T cells. Starting with CD1 proteins. Well, CD1 protein has 5 isoforms. That is A, B, C, D, and E. And these differentiation proteins are not expressed on T cells, but are found majorly on thymocytes, that are immature T cells, and dendritic cells, but are involved in presentation of lipid-containing antigens to the T cells. These CD1 molecules are structurally similar to MHC class I molecules. As, just like MHC antigens, they form complex with beta-2 microglobulin. And so, just like MHC molecules, they can present antigens to T cells. Mycobacterial lipidic antigens can also be presented by CD1. After CD1, next is CD2. This CD2 is found on the surface of T cells and is known as the early T cell marker, as it originates on all T cells' surface in their early developmental phases. This CD2 serves as the receptor for CD58 that is found on other lymphatic cells. In this way, it helps in cellular communication of T cells with other immune cells. Then comes a very important CD protein, that is actually CD3 protein complex. This CD3 is especially important because of its co-stimulatory action along with TCRs. This CD3 is actually a cluster of proteins, which consist of a number of important membrane-based molecules that are closely associated with T cell receptors. And they help them in intracellular signal transduction. This CD3 complex usually made of 4 different proteins that are gamma, delta, epsilon, and zeta. This CD3 complex consists of two heterodimers. I am showing as 1 and 2. Here the first one consists of gamma protein and epsilon protein, and the other one is made of delta protein and epsilon protein. Then beside TCR, two zeta proteins linked with disulfide bond makes a remaining portion of this CD3 complex. The primary function of this CD3 complex is to transmit signals, that is signal transduction, from the TCR to the interior of the T cell upon recognition of antigenic peptides presented by major histocompatibility complex (MHC) molecules on antigen-presenting cells (APCs) or infected cells. Upon antigen recognition, by binding this antigen MHC complex, the CD3 complex is responsible for transducing the activation signal to the interior of the T cell. I am not going to explain the detailed mechanism here, for that you have to watch another video under the title T cell activation. Here I'll explain it briefly. Actually, what happened is that these CD3 proteins contain special sites called immunoreceptor tyrosine-based activation motifs, or simply called items, within their cytoplasmic tails. These items then become phosphorylated upon TCR engagement with antigen MHC complex. Phosphorylation of these items in the CD3 complex leads to the recruitment and activation of various intracellular signaling molecules. These signaling molecules then initiate a cascade of events, triggering the activation and proliferation of the T cell. So, we can say mainly these CD3 molecules are involved in intracellular signal transduction, along with TCRs. And additionally, this CD3 complex is involved in the proper expression and surface localization of the TCR. So, overall, the CD3 complex serves as a crucial link between the TCR and intracellular signaling pathways, enabling T cells to detect and respond to specific antigens. Now, after CD3 complex, next important CD protein is CD4. That is another viral differentiation protein, and is expressed predominantly on a particular subset of T lymphocytes, called helper T cells. This CD4 interacts with major histocompatibility, complex class II or MHC2 molecules and contributes to antigen recognition and immune responses. CD4 serves as a coreceptor for the T cell receptors, enhancing the sensitivity of T cells to antigen presentation. It enhances the binding of the TCR to the antigen MHC2 complex, leading to stronger and more specific T cell activation upon antigen recognition. And they do so by interacting with coreceptors on APCs, such as CD40 and CD80. These interactions provide additional signals for T cell activation and modulate the immune response. Other than this, the CD4 also serves as a receptor for the human immunodeficiency virus, HIV. The virus binds to CD4 on certain immune cells, such as helper T cells, leading to infection and depletion of these cells, which contributes to the progression of AIDS. Now, next known CD antigen is CD8. That is just like CD4, expressed primarily on particular subset of T cells. And here, that subset is cytotoxic T cells. Upon the surface of cytotoxic or killer T cells, these CD8 molecules interacts with major histocompatibility, complex class I or MHC1 molecules, playing a role in cell-mediated immune responses. This interaction helps stabilize the association between cytotoxic T cells and target cells, facilitating the recognition and destruction of cells displaying foreign antigens. This CD8 also acts as a coreceptor alongside the T cell receptor, just like CD4. But it is present on cytotoxic T cells, unlike CD4, that are found on helper T cells. It enhances the binding of the TCR to the antigen MHC1 complex, leading to stronger and more specific T cell activation upon antigen recognition. Also, CD8 expressing T cells can respond to cytokines, such as interleukin-2 and interleukin-15. These cytokines promote the survival, proliferation, and effector functions of CD8 plus T cells, enhancing their cytotoxic capabilities. So, we can say that CD8's involvement in immune cell interactions is crucial for immune surveillance, defense against infections, and immune regulation. So, these were some famous and more authoritative CD proteins associated with T cells. But there are many others. As we mentioned, there are more than 370 CD proteins are known yet. Such as the CD5 antigen and CD7 antigen are also found on T cell surface, where CD5 is involved in signal transduction, and is not only found on T cells, but are expressed in a subpopulation of B lymphocytes as well. And CD7 antigen is one of the earliest T cell markers, just like CD2. But the mode of action of these antigens is still unknown. Another known CD protein is CD28. It is expressed on the surface of T cells, where it serves as a co-stimulatory receptor. As it interacts with CD80 and CD86 molecules on antigen-presenting cells, providing a necessary co-stimulatory signal for T cell activation and proliferation. Another one is CD25, also known as the interleukin-2 receptor alpha chain, and it is upregulated on activated T cells. And this CD25, once activated, plays a role in T cell proliferation, survival, and immune regulation by binding to interleukin-2. Another CD protein expressed on T cells is CD154. That has another name, that is CD40L, which means it acts as a ligand for CD40 protein found on antigen-presenting cells and mainly B cells. Or simply, it interacts with CD40, providing co-stimulatory signals necessary for B cell activation, antibody production, and immune responses. So, this was all about the cell surface differential molecules that are associated with T cells, or simply T cell antigens. We can't discuss all of them, but here we tried to cover all the main and important T cell antigens, such as CD3, CD4, CD8, and some relatively less important ones, such as CD5, CD25, CD28, etc. These CD proteins are integral to T cell biology and function, contributing to various aspects of T cell-mediated immune responses, such as they regulate T cell activation, differentiation, and effector functions, ensuring the proper functioning of the adaptive immune system. We will discuss the detailed functioning and mechanism of action of these T cell antigens in our next video. For now, this was all about the T cell antigens or cluster differentiation proteins. For more such interesting and full of knowledge videos, keep visiting sqadia.com. Ever felt overwhelmed by the traditional way of learning medicine? Enter the future of education with digital learning. Whether you're refining your knowledge of biochemistry or immersing in cell biology, it's all there. Understanding complex medical subjects has never been easier or more efficient with engaging, interactive content. Welcome to the new age of medical education. Learn anywhere, anytime, at your pace. Powering your medical journey. sqadia.com